Alright, ladies and gentlemen, this is our final video for chapter 11. It's our 7th video. Good God, I'm so sorry. And we're going to go from, we're going to do a quick review on DNA, helping to lead and build a protein. We're going to watch a new little model activity for that. And then we're going to dig right into problem 3 and 4, which is you can change the proteins the DNA codes for by changing the DNA code. So when DNA code gets messed with, damaged, such as from all the chemicals in our environment like pollution, the lifestyle we have, maybe drug use or alcohol abuse, or even sun damage, it all can potentially change the coding of our DNA. And if it damages or changes the nucleotides of DNA, you got yourself a mutation. So we want to see and kind of look into not only what causes mutations or changes to DNA, but what does it really do to us? What's the consequence, right? Possible illness, whatnot. So we really just want to discover more about that as well. All right. So without further ado, let's do a real quick recap on how DNA makes proteins, and then we'll start looking at what happens when you mess with DNA. What does it do to that protein? I mean, what do mutations do to proteins? So let's do it. Just as a recap, we have our chromosomes made of DNA and our nucleus of each of our cells. And so within each of those hundreds of trillions of cells that make us, that DNA code in there is just ready to make tens of thousands of different proteins. Here's a particular gene. We're not sure what this gene codes for, though. We're not sure what protein it makes, but we do know that the DNA is going to be turned into RNA. So let's watch it. It's going to be made into RNA. You can see RNA at the very top. So the DNA opens up, RNA is made. This is RNA polymerase, by the way. I think you know that. Alright, this kind of gives you an idea of how the mRNA at the top, and we know it's RNA because it has U's, the U nucleotide, right? So it shows you how the DNA nucleotides partner up with the RNA nucleotides to make that RNA code. Why DNA goes to RNA, I don't know. It's just how our body functions. Let's see what happens with that RNA now. Not everything looks the way we're used to, but you got a tRNA or a transfer RNA, and it's carrying an amino acid. It's going to go straight for, and it's kind of cool how it draws this one, but you got the RNA code made from the DNA, and every 3 RNA nucleotides are called codon, and you can see how they pair up. So check it out. Each one of the tRNA is bringing amino acids. Now it's just bonded 1, 2, 3, 4, 5, 6, 7 different amino acid molecules together. And let's check out what protein it makes. So it's a straight one-dimensional chain of amino acids now, but let's let it fold together, and it makes a complicated protein we call insulin. And insulin is really important when it comes to your body absorbing sugar into its cells. So no cell of your body will absorb any sugar and feed itself or make energy from it without this protein insulin telling it to do so. So insulin is a special chemical messenger that tells cells exactly what to do. So this protein, important job. Your cells would starve to death if it wasn't for this protein to tell cells, hey, get some sugar. Now let's think about it. What happens when the DNA is changed? What if we take a single nucleotide and change it, or just delete a whole section? Well, I know the images are small, but what I want you to do is, in your notebook, for slide 42, it says, make a hypothesis. So write slide 42, and then you can put my hypothesis is, and then use these words nucleotide, DNA, and phenotype to describe what's going on in any of these images. You have this picture showing nucleotides. This picture also shows nucleotides being changed by X-rays. So X-rays or light is hitting the animal, and it seems to be changing the nucleotides. You have phenotypes, 2 different phenotypes, 2 different fish. Their phenotypes are different, presumably because of a mutation or a change to their DNA. One has scales, one doesn't. Then you have a lot of phenotypes of dogs, starting with the wolf. You've had all these phenotype changes of different dog breeds from the wolf. use the word nucleotides, DNA, and phenotype to try to explain what's going on in any of these pictures based on genetic changes. Let's look at this animation. It's one of our YouTube channels we watch a lot called Clearly Stated, or Stated Clearly. And you can see there's DNA here. It's T's, C's, A's, and G's. And let's look at what they call a single point mutation, or a change to the DNA. And this is a gene that they found in certain species. The gene is wonderfully named FGF5. Let's see where they're going with it. Pay attention to the subtitles. And let's see what we can make clear. So there is a specific single nucleotide that's going to identify it, but there's been a single change to this gene called FGF5. And this is, so it was changed right here. It was a G and a C, and it was changed to T and A. And it explains the whole difference between hair lengths in dogs. So you change those one nucleotide in DNA, you mutate that one nucleotide in DNA, and you get dogs of different hair length. Pretty interesting. Now here's another interesting thing, is there's a toad, right? And it's in nature, and it produces a toxin. It has a gland here, so it produces a special toxin or protein inside itself. Now, most organisms, they have these little channel proteins, and they pump things in and out of each cell. So all the cells of any organism need to bring in good stuff and remove bad things. Well, the toxin of this frog actually messes with. This is a protein, that's the toxin or the protein, and it keeps your channel proteins from doing anything. If an animal in nature bites the frog, or sorry, the toad, they'll get this toxin inside their body, and it'll attack the proteins of their cells. It'll stop these channel proteins from working. And if a cell can't transport materials in and out of its through its cell membrane, if it can't use its channel proteins or its transporter proteins, this toxin basically will kill the cell. And so can you imagine billions of this protein? So you bite into the toad, right? Say it's a wild animal, you know, a snake bites this toad. It gets billions of this protein released into its body, and all those billions of this enzyme bond to the channel protein or this transporter protein and basically kill a bunch of cells in this organism. That's how toxins work. These toxins often can be proteins, like the toad makes, that shuts down some important cell function. They've found that a lot of species have a mutated version of the protein, and check this out, the toxin can't bind to the so there's lots of species in nature that have adapted by mutating their. Apparently those species that are surviving, their mutated version of the protein was due because their DNA changed. So a lot of those species in nature, they changed their DNA, they changed their protein, and now they're immune to that toxin. That's pretty cool. And that's why the hedgehog's so happy. Its DNA got changed, has a healthier protein, it's good to go. What kind of things can change DNA? We know the mutation we just saw protected some species from the toxin of the toad, but sometimes mutations happen we don't want. Sometimes mutations happen based on our lifestyle, things we've done to ourselves that can cause harm. Let's check it out. One thing that can damage DNA is environmental pollution or cigarette smoke. So anything that we do in our lifestyle can have real harm to the DNA. Other chemicals in the body, even hydrogen peroxide our body produces, and it can damage and mutate the DNA. The thing is that our body's made up of trillions and trillions of nucleotides. Each chromosome contains billions of nucleotides. And even if just one base is damaged, it can have huge effects. So even just one nitrogen base gets altered. It can have a really big effect. Even just one. So if even just one nucleotide is wrong in there, it can cause a big issue. UV light is also one of those big things that can damage the DNA, so we're always told to wear sunblock, and it's really because if we go out there and we're not wearing it, it can really damage the DNA too. So the environment kind of is out to get us. They can damage our DNA from pollution to the sun exposure, all kinds of things. Stress. If you're stressed in life, even stress can mutate your DNA. Here's a great video from National Geographic, and it shows how your DNA can actually start unraveling. Let's check it out. So if you're too stressed, you'll actually wear out and damage certain sections of chromosomes or DNA due to stress. From drugs to stress, to the sun, all kinds of things can change our DNA and potentially do us harm. And then, like in another example, sometimes DNA changes and it was beneficial, helps the species survive. Slide 43, please write that in your notebook and create this brainstorm web here, and it says causes mutations. So try to list a few things that can actually lead or cause your DNA to mutate. Go for it now, please. Here's slide 43 if you want a little help. And of course, who knows, things like high temperature can damage DNA. Every time you cook your meat or cook a plant, anytime you cook any food, you're damaging its DNA or mutating it. Radiation from the sun or chemicals, chemicals in general, household products and drugs and alcohol. Or mutations can happen naturally. Anytime your cells are reproducing, your DNA has to get replicated. When those chromosomes and DNA have to get replicated, mistakes happen, and it can lead to a mutation that causes a different protein also. Alright, here's slide 45. Slide 45 isn't done in your notebook, but it does cover an activity we'll be doing in class. If you missed the day we did this in class, please watch this video, and it's a little quick reference. So the idea is what happens when DNA is damaged or changed? So we're going to cut to this simulation. I'm going to show you a little bit of observations you can come up with when you need to do this, in case you missed. Alright? So we're going to try to learn more about how DNA is damaged and changed, and really what is the result of that, what happens when it's changed. Alright, so we have our DNA here, and if nothing happens, if everything's normal, it gets transcribed to tRNA. This tRNA now, sent with the red border, and then you take the RNA, and you make, from the RNA and the codons, you make amino acid chain, you got yourself a protein, right? Wonderful. Well, you can also reset it, and you can click on any of these nucleotides. Substitution means to change, say, or mutate a T to an A. So you can change what was a T nucleotide, make it an A. Now, if you then transcribe the DNA to RNA, that T was changed to an A, which is going to result in a new amino acid code. And the second amino acid now is different. So technically the protein overall is different because it now has a different amino acid that makes it. Now we don't know the function of this protein, but we know we changed the protein. The mutation had an effect on the protein. It changed the protein because we changed the DNA nucleotide. Another thing you can do is you can just delete. So this T right here, before we changed it, you can also just, you can add an A. Say there was never, say the gene was made up of 100 nucleotides. Well, now it's made of 101 because we just added a new nucleotide in there. And that's going to have effects on the protein as well. Yep. And given that we added a new nucleotide, which is a mutation, it's made a protein that is certainly different and probably dramatically different than the original protein from that gene. You can basically change a nucleotide, delete a nucleotide, or add a new nucleotide. And this can happen anytime DNA is damaged or changed. Anything from the environment can do those things, or your body can just do that automatically when cells divide and a mistake is made. But you get the simulation, right? You write all your observations down, and then you tell me what you learned. You state what you understand, or you'll draw a model to represent what happens when the DNA is changed. You can talk about all the different ways the DNA can be changed, from a substitute mutation to a deletion or adding a new nucleotide. And you just go for it. Alright, good luck. Alright, we'll have one more video series to cover our last 3 or 4 slides, so have a great day and Look forward to video number 8, I believe.